Many of you, I am sure, are familiar with paper-based lateral flow test devices. The most commercially successful of those is the pregnancy test, shown on the right side of the figure. There are many more such test strips in the market for the detection of drugs of abuse, gluten in food, and many more. A typical structure of these devices is shown on the left side. On a plastic sheet, first there is placed a cellulose sample pad, followed by a membrane constructed of glass fibers. This is the conjugate pad, in which detection antibodies tagged with gold nanoparticles are dried. Following that is a nitrocellulose membrane, on which, on the test line, are immobilized capture antibodies, and on the control line are immobilized antibodies specific to the detection antibodies. Typical dimensions for these test strips are 5 millimeters wide by 5 centimeters long. In operation, a biological sample in fluid form is deposited on the sample pad and begins to flow by capillary action through the conjugate pad. The antigens in the sample, if there are any, conjugate with the detection antibodies, and this conjugate then proceeds to flow over the test line, where the antigens are captured by the capture antibodies, thus congregating the nanoparticles, and a visible color develops. The test strips are easy to use, they are autonomous, they are low cost, and they're highly portable, and do not require any power for their operation. Their limitation is that they can only handle one reagent, so they cannot perform more complex assays. Polymer-based lateral flow devices overcome this limitation because they can manipulate the flow of a number of reagents. A typical architecture of such a system is shown in this figure. The system consists of a plastic disposable microfluidic chip which contains a number of microchannels within it. In these channels are stored the various reagents needed for a particular protocol. The slugs of reagents are separated by air gaps. In the center of the chip is located a test site that is functionalized with capture antibodies. After the sample, in liquid form again, it is loaded into an input port. The chip is inserted into a box containing a number of electromechanical valves, a pump, an optical sensor, a microprocessor, a radio receiver and transmitter, and other electronics. The operator then starts the test through a command on the box or via a smartphone, and the various fluids are loaded to and pushed out from the test area in sequence according to a predetermined protocol. When the test is completed, the box communicates the results back to the phone or presents them on its display. Like the test strips, these devices are autonomous and portable. However, they have not been used successfully commercially yet, and this may be due to the fact that they require the operator has some training in order to use them. They do cost more and need power to run. The lateral flow device I will present today combines the ease of use and low cost of these paper-based test strips, but can manipulate several reagents so that, like the polymer-based systems, it can run more complex assays autonomously. A top view of one of our devices that can handle the 3 reagents is shown in this slide. It consists of a central channel that is similar in construction to the test strips I described previously. There is a sample pad, a conjugate pad, and a nitrocellulose membrane, and a waste pad that serves to absorb all the fluids. The device, however, has 2 additional inputs, one on the left and one on the right of the central channel. In these pads may be stored 2 reagents, whose flows are triggered by the sample fluid in a predetermined sequence. This short video demonstrates the operation of the device. In this figure, you will see the same device as before, rotated 90 degrees. First, the 2 reagents are loaded in the 2 pads and remain there until triggered by the sample fluid. The sample fluid will be pipetted onto the sample pad and you will see it flow down the central channel. After some time, the red fluid will begin to flow, and sometime after that, the dark green fluid will flow. The video is sped up 25 times. Its actual duration is about 12 minutes or so. You see the two reagents have been preloaded and are not flowing. Now you see the pipette reaching into the sample and loading a clear fluid onto it. This clear fluid begins to flow down the central channel by capillary action. You see that it is well past the test spot, and now the red fluid begins to flow and pass over the test spot. Now the green fluid does it too, and passes over the second spot. Now I will play the video, but I will not talk over it. In this demonstration, the fluids are all DI water with food coloring. The reason the reagents remained on their respective input pads and did not flow until triggered is because beneath each one of them is a fluidic diode, or valve, fabricated in paper that blocked their flow. In the left side of the slide is a simplified cross-sectional view of the fluidic diode. It consists of two layers of paper, separated by a double-sided tape. In the tape is punched a hole, and in that hole there are two discs, both made out of paper. The hydrophobic disc has been soaked in a solution which renders its fibers hydrophobic, but remains porous. The hydrophilic disc is infused with tween 20 surfactant. A symbol of our paper-based microfluidic valve is shown to the right side of the slide. In operation, if the fluid approaches the diode from the forward direction, it dissolves the surfactant, which goes into the hydrophobic area and renders the fibers hydrophilic again. If the fluid approaches from the reverse direction, it faces a hydrophobic region and cannot penetrate it. However, unlike electrical diodes, once the diode is bridged, it now allows fluid flow in both directions. So, the reason the red and the green fluids in the video could not flow was because underneath each pad was a diode, as shown in this figure. There is an additional important and critical detail shown in this figure. The black blocks around the red fluid represent wax. In real devices, this wax is deposited on the paper in the desired pattern by an inkjet-like commercial printer, and then the paper is heated to drive the wax through its thickness. This forms a solid hydrophobic barrier. This is the reason the red fluid in this figure is contained and does not spread laterally. The other point I made while showing the video is that the reagents are triggered by the sample fluid. This is demonstrated in the figure. At time equal to 0, a drop of red fluid is loaded on the top or reverse side of the diode and remains there. From the bottom side, a green fluid approaches, which dissolves the surfactant and begins to render the hydrophobic region hydrophilic, allowing the green fluid to penetrate throughout the diode and bridge it, thus triggering the flow of the red fluid. The two mix, resulting in a yellow color. The next step is to demonstrate that the device can autonomously conduct an assay that requires more than one reagent. For that, we selected to do an enzyme-linked immunosorbent assay, or ELISA. In this assay, the antigen is rabbit IgG, dissolved in buffer. Detection antibody is monoclonal mouse anti-rabbit IgG, tagged with enzyme alkaline phosphatase. The capture antibody is in a monoclonal mouse anti-rabbit IgG, and the substrate is BCIP NBT. The first step is to immobilize the capture antibodies on the nitrocellulose membrane by placing a drop of solution containing the monoclonal mouse anti-rabbit IgG and letting it dry, thus forming the test spot. This is followed by a blocking step that is performed on the nitrocellulose membrane, the conjugate pad, and on the input pad to prevent nonspecific binding, as well as to prevent the loss of the antigen. As the sample flows past the conjugate pad, the antigens conjugate with the detection antibodies and flow past the nitrocellulose membrane. The wash solution is activated next, and it flows past the test spot and the rest of the nitrocellulose membrane to remove any of the detection antibodies with ALP enzymes that are not already attached to a capture antibody. Finally, the substrate is activated to flow. It interacts with the enzymes immobilized at the test spot, producing the colored precipitate, which is detected. In this slide, I am showing one of the chips we fabricated to conduct this assay. In the right side is a top view of the chip, and is similar to what I have shown previously. On the left side is an exploded view of the device. It contains 2 layers of filter paper, onto which channels have been defined by the wax. The black regions are where the wax has been deposited and melted into the paper. The white regions are the channels and consist of virgin cellulose paper. There are 2 layers of double-sided tape. In the middle tape layer, a number of holes have been made. The 4 in the center is where the 4 diodes are formed, all facing upwards. The small hole in the back is filled with cellulose paper and serves to form a short between the sample pad and the top layer, and the delay or trigger channels in the middle of the paper. The square hole in the left side forms another short between the waste pad on the top and the blotting paper on the bottom of the stack. It serves to absorb all the fluids. The top paper also has 2 cuts in it. One is covered by the conjugate pad and the other by the nitrocellulose membrane. The arrows in this figure serve to illustrate the fluid flow. I have drawn an equivalent circuit diagram of the device to explain its operation and how it autonomously conducts the immunosorbent assay. In this slide, the lines represent channels, like wires in an electrical circuit, where fluid may flow. As I mentioned earlier, in the conjugate pad, we have dried the detection antibodies, which are tagged with ALP enzymes. These antibodies are free to move. In the nitrocellulose membrane, we have formed a test spot, which consists of immobilized capture antibodies. To conduct the assay, first, the wash solution is loaded onto the wash inlet, and the substrate is loaded onto its respective inlet. Both of these solutions remain in place, as pointed out earlier, because beneath each of them is a diode, V1 for the wash and V3 for the substrate, that prevent their flow. When the sample is loaded onto its inlet, some of it flows down the central channel and over the test spot, where it picks up the detection antibodies and brings them over the test spot to form a sandwich. Note that diodes V2 and V4 prevent the sample solution from flowing towards the wash or substrate inlets. Now back to the sample inlet. You see that a portion of the sample flows in delay channel 1. When this fluid reaches diodes V1 and V2, it bridges them, allowing the wash to flow towards the central channel and over the test spot and remove any excess detection antibodies tagged with ALP. The delay channel, as you can imagine, is drawn long enough so that the sample fluid has time to pass the test spot before the wash is activated. After the sample fluid and wash have passed the test spot, a portion of them flows into delay channel 2, where it turns on diodes V3 and V4, now allowing the substrate to flow over the test spot and create the colored precipitate. In a final device, the housing holding the device will also have receptacles into which the wash and substrate solutions will be held, so that the user will only have to load the sample in its inlet for the assay to begin. Using this device and the protocol described above, we have obtained a dose response or standard curve for our system. The curve is shown at the left. The limit of detection was about 4.7 nanograms per milliliter, which is quite respectable. The right side shows 2 chips at the end of the assay. In the top one, the test spot was obtained for an IgG concentration of 0.1 micrograms per milliliter, and for the bottom one, the IgG concentration in the sample was 1 microgram per milliliter. The darkness of the spots corresponds to the signal, and it is seen that there is quite a difference between them.